Yeah, so as well as TB and why it's complicated, we'll go through a little bit on how to set up a laboratory. And again, I think we've kind of gone through some of that already, so we can try and speed through it. But in terms of setup, what we found generally that, so with lead times and delivery times, some of the kits have quite a long lead time. So they release them in sort of waves. Then the flow cell, for instance, the R9 version, which is currently generally available, you can get that relatively quickly, but the R10 version, there's sort of a waiting list because it's a newer technology, and people are kind of testing it out. Nanopore is very much the community is testing how well these things work. So they have sort of a system of, this thing is completely tested. We are fairly confident this is how it works, all the way through to, we've just released this version. So, you know, what you're finding is actually validating what we think. So some of the newer kits take a while to come. The website suggests a working week for orders to arrive to what it describes as the rest of the world. And I think that's anywhere outside of Europe. Because my lab where I am in London is not very far from the factory, things tend to arrive once I've actually physically placed the order within a couple of days. But as John and I know, trying to get things in and out of countries can be more problematic. And I'm sure probably a lot of you have already dealt with getting things in and out through customs and whatnot. So customs issues are probably going to take a long time. They will arrive. Usually everything arrives not frozen, but refrigerated, even the kits, don't they, I think? So if your customs time is really long, it's something to maybe consider. The flow cells, and I cannot stress it enough, should not be frozen, because if you freeze them, all of the pores will pop and you will have wasted a flow cell. It won't work at all. But the kits, most of the kits should be kept at frozen, but they will often arrive at room temperature because it saves on delivery costs. But customs obviously can take 2, 3, 4, sometimes more weeks. I was working in Uzbekistan and it took 3.5 months to get it through customs. So just something to consider when you're ordering things. But Nanopore generally are very responsive when you kind of contact them to get things sorted. That's generally what you found, is it, on this end here? In terms of the reagents, as I say, often they send the kits out in a refrigerated box. It's sort of packed with lamb's wool generally. But once you receive it, they should be kept in the freezer. But check on the boxes where, you know, what the temperature is. And as I say, don't freeze the flow cells. But they have created a field sequencing kit that doesn't need this cold chain. So if you are going out into the field and plan to do sequencing sort of out in the wild, as it were, then they do have a kit that actually doesn't need any kind of cold chain. And I know that they did sequencing in, I think it was Antarctica, and they just used a mug of coffee to get it up to 80 degrees. So they just stuck everything in like hot drink, and that's how they did it, rather than use PCR machines and things like that. So it very much does have an option for kind of very rural, if that's something that you are interested in doing. The flow cells, so as John said, they arrive with this storage solution in that kind of protects the pores. And if you keep them unopened at room temperature, you can keep them for 4 weeks after the delivery date. But if you keep them in the fridge, they can be stored, well, the warranty, I think, Nanopore's warranty is for 12 weeks. I found a flow cell in our cold room about 4 years later, and actually it was really good. So it just depends how you store them. Sometimes they're fine, but actually sometimes they go down and their active pores drop for no apparent reason, just sitting quietly in the fridge. So it's probably better to aim to use them all up as quickly as possible. So if you need to think about your timings and how quickly things come and how much you might be doing, it's cheaper to buy them in bulk. But at the same time, if you're then not going to use half of them for a year, then you might find that actually the number of active pores kind of lowers to the point where you probably would have been worth just buying a separate section, separate group. You also need to QC the flow cells. Nanopore suggests within a couple of days of them arriving, because when you QC them, it will give you an active pore number. Generally, this should hopefully be somewhere between about 1,600 to 1,000, maybe 200 or 300. If it's anywhere below 800, then that's below Nanopore's warranty, and they should, in theory, send you a replacement one. I got one once that was 1,850, and I was really excited because that's the best flow cell I've ever got. And obviously, the more active pores you've got, the more sequencing will happen on that flow cell because you've got more pores doing more sequencing. So the more pores you've got, the better it will work. Potentially, if you have a low number of pores on your flow cell, so we QC'd ours yesterday. Some of them, I think one had 1,400 and the other two had about 800. Yeah. So over time, they have decreased. So what we might do with the 1,400 one, we might put on, say, 10 samples. And the ones that are lower, we might put maybe, I don't know, 6 or so on. Because obviously you have to, it's not an exact science, but if you try and put the same amount on to half the amount of pores, then obviously you're going to get half the amount of data for each of your samples. So you just have to sort of bear that in mind. So in terms of throughput, so the nanopore, if you're doing whole genomes with this new kit, and it's sort of a simple genome that you're looking for, then we have been able to run about 24 samples. If you are looking to do less than that, it might be worth buying a flongle. You can see that the flongle, that's the flow cell, that's the kind of adapter, and then it just fits onto the MinION itself. So you can run maybe 2, 3, 4, 5, however many you want. It just makes it kind of cheaper overall. They are only guaranteed for 4 weeks after the shipment date rather than 12 for MinION though. So you would have to potentially use them quite quickly. Power issues. You need power for the duration of your run, and your run might be up to 72 hours if you're doing whole genome sequencing. So if your power cuts out regularly, you would probably want a UPS. It might be worth, if your power is dodgy and a UPS is not sort of possible, worth buying a laptop rather than a PC, because your laptop will have at least a bit of battery to run on backup. The Mark 1C doesn't survive if a power cut happens, though, does it? So it just cuts out, right? Yeah.
The Mark 1C now.
So if so if power is an issue,
like a UPS,
yeah.
Like
as I say, if the power cuts out and the computer cuts out, your sequencing will stop, and it might be quite difficult to then start it up again.
Start to see.
Yeah. So invest in a UPS or maybe a laptop for your sequencing might be a better option. Yeah. The internet. So we've got different, I say that because as I think I've already told you in my last workshop, it was a problem. With the MinION, so the little one, that needs the internet to start up the run especially, and then it pings to the Nanopore server occasionally to send data to and from it. So especially at the beginning, you definitely need a fairly sensible data connection. As I told you, I ran a workshop in Tanzania in August, and we had internet, and then it went away, and everybody was running around trying to find Wi-Fi, trying to find an internet hotspot. We got it to work after the 6th attempt, but it was a lesson learned for me. You can ask Nanopore nicely, and if you have a good enough reason, they will give you an offline version of Minno, which is the software that you use to run it. Alternatively, you could buy a Mark 1C, which, as John said, is essentially the MinION, but it's got a little computer attached to it, and it's all completely offline. You don't need any internet to run the sequencing, but you do need internet to update the software, as we found out. So at some point you will need to update it.
Like a year or so, it will stop working and it won't let you run anything until you update the software.
It's actually also really important to keep the software as updated as possible, because the programs get more and more accurate. And so, as I was saying before, if you've got some samples that you've done already, using the new version of Guppy to then rebaseball is actually, you get much more accurate data. So keeping your Minno software or Mark 1C version as up to date as possible will give you essentially better results, or at least more accurate results.
Yeah, that's fine.
How do you get the data?
You mean from the MK1C? So the MK1C stores all the fast 5 and the fast Q data on board. So when you're done with the run, you can demultiplex and barcode everything on the Mark 1C itself. Yes, and then you just pull off the fast Q files.
It's like a little computer in its own right. Attached.
So Yeah, you can connect the USB because there's a port on the side that you can use. You can connect it, and then you can transfer files to a hard drive or a USB stick. But I find that it's either probably better to connect it to like a network, right? So you can get the data off it a bit quicker. With the port for the USB sticks, it's a bit slow. So I think it's only good when I'm getting off FASTQ files. But if I'm trying to get off FAST5 files, it will tell me like copying will take 3 hours or whatever it is, which is like not. So if you're going to take off the FAST5 files, better you have it connected to a network, so you can just go in, get your FAST5 files, and then do your high accuracy based calling if that's what you want to do. But for fast based calling, you'll find the FASTQ files at the end of your run. That's fine.
It's got, is it a terabyte of space on it?
Yes, it's got 1 terabyte worth of space. So
more than enough for at least one run.
Yeah.
But yeah, I would recommend moving then that off to do what you
either
want with
move
it
it off or like, move it off and then delete what you have so that you can.
Because if you don't delete or if you don't have enough space, then it will just stop the run. It will save what you've got up until that point. But if you've got a run of that's going to generate 70 gigabytes worth of data and you've only got 20 gigabytes of data space, then it will just stop the run and then it won't continue. So you would essentially kind of half to the use of your flow cell. So I think actually when it starts up sequencing, certainly the new versions of Minnow actually say you have X amount of storage space, this isn't enough. It often massively overestimates because sometimes it says you have 300 gigabytes, it's not enough. But once you've done some runs and you know roughly how much you're going to get, you will know how much space you'll probably need. Yes. A lot of the analysis programs as well can use internet access. So we're going to have a go at TB Profiler on Friday. There are 2 versions of that. There's a command line based version, and then there's a cloud sort of browser based version where you sort of upload your samples to the database. That is so 1 or more of your samples, they can be sort of gigabytes worth each individual sample. And if your internet isn't particularly strong, that can take ages to upload. So with things like TB Profiler, that it has 2 versions, it might be that actually the command line one is worth your while learning, because if your internet isn't powerful enough, you can do that all sort of offline. Conversely though, if you don't have a particularly powerful computer and you have good internet, like for instance, this isn't a hugely powerful computer and the internet at UCL is very, very powerful, it's much quicker for me to upload it to the cloud than it is for me to run it on the command line. So yeah, it provides the same output, but it's just a different way of doing it. And a lot of the kind of user-friendly graphical user base, so buttons rather than command line versions of programs are all kind of online. So internet access is potentially quite important, and it might be worth investing in a better internet specifically for sequencing maybe. And also storing data as well. So as I said, at UCL, we have these research storage drives. They're all based on our kind of network and our server. And so I can only access them if I've got the internet. So that's also where you store data is also something to consider, and whether you have enough internet to kind of upload 200 gigabytes worth of data to a folder on the cloud. And I think I mentioned offline configurations. So yeah, a mid-ion flow cell can generate as much as 50 gigabytes, although I'm finding with metagenomic data that's actually much more. So storage can definitely become an issue. It's worth keeping your actual sequencing computer or Mark 1C as free as possible. So I would do one run, remove all the data, so that is free to do the sequencing. Otherwise, you're kind of clogging it up. And as I said earlier, running out of disk space will just halt your sequencing experiment. And unless you have a GPU, so most computers run on CPUs, so all of our laptops are likely to run on CPUs, which is central processing units. They are kind of what the computer uses to process all of the data. A GPU is a graphical processing unit. These arose from video gaming. So these processing units are really, really good at dealing with large amounts of small data at once, because they were originally used for processing graphical, video games. And obviously, as you're moving through a world, you're seeing all sorts of things and it's changing that scene every millisecond or whatever. actually GPUs work really well for sequencing because you are also processing billions and millions of tiny bits of data at once. So if you have the capacity and the funds to buy a GPU-based computer, which we're going to try out, then I thoroughly recommend that. It depends what you're doing though. If you have a fairly powerful CPU computer, so this is a 32 gig of RAM CPU laptop. If I'm running sequencing, I might find that if I'm trying to base call at the same time, so you can turn base calling off if you want. If I'm trying to base call at the same time, it won't be able to keep up, so it won't be base calling in real time, so it will lag behind. So if you have a GPU, that's just going to be automatically, as soon as it's sequenced, it will also base call it, and so you'll have that data. So if you have a smaller computer, you might want to just turn off base calling because it takes up valuable kind of computing power. So it's just something, again, to consider in terms of time. So again, if you can afford a GPU, and I think you can add GPUs to existing computers. I think they're about £2,000-ish, so about $2,500-ish each. So even if you have a, even if you have a computer already set up, it's possible to sort of install it and make it more powerful. But as I said earlier, definitely the most powerful computer you can get is very much going to save you hassle in the long run. And those are just the specifications for the Mark 1C. So a terabyte of storage, 8 gigabytes of RAM, which sounds small, but actually it's the GPU that's really important on that. And in terms of data storage, it's also really important to consider human sequence data if you're storing it anywhere. So GDPR, so data protection. If you are doing, say, direct from sputum samples, a huge proportion of that is going to be human DNA because it's a human sample that is going to get extracted along with your TB or whatever you're looking for. You can deplete the DNA before you start. So in the lab, you can actually deplete the human DNA, but you do also have to consider if you're uploading it to somewhere, if you're uploading it to a program like, say, TB Profiler, you have put that human DNA data into the cloud. You don't necessarily know where that server is. Some countries have less strict rules on how they process that data. In the UK, for instance, we're not allowed to store data in certain countries because their rules on GDPR are much lower than ours. So it just depends where you are and what you want to do with it. But I wouldn't recommend, I would try and remove as much human DNA as possible, because otherwise you might come into problems, because if that data got into the wrong hands, then they could see all sorts of things in a human genome that shouldn't maybe be seen. This is just a quick run through kind of the whole pipeline of what we've been talking about so far. And the main point, as I've been saying, is that it's for TB. We're trying to make it sort of pragmatic so that it's cheap enough to work, but also easy enough for people to use. So what our kind of original problem was that can we use whole genome sequence to complement routine diagnostics? And I think, as Keith has pointed out very much, yes, that's something that people would really like to do, especially in really hard to treat cases. So people who have not responded to their treatment, is it that they have relapsed? So is it the same organism, or is it that they've been reinfected whilst they've been back in their community? So as we were saying, the SNP difference between the same organism or a new organism can be anywhere as little as 10, 13 SNPs. So that's a really small number, and actually the only way really to identify to that kind of level is whole genome sequencing. And then looking at drug resistance, as Keith has said earlier, it's quite a complex scene. There are various SNPs for various different resistances, and the more data that we get, the better we will be able to identify those resistances, and therefore better pipelines for further down the line. Yes, balancing the cost, accuracy, because it can be really, really cheap, but if it's not very, very good, then there's no point. And then actual feasibility in the labs. This is the pipeline itself. This is kind of all the way along. So we start with the growth and extraction, because at the moment this is based on culture. What we're trying to balance is the speed of the extraction versus the concentration, molecular weight, and quality, as we mentioned in the previous slides. But equally, CTAB is a painful method. It's very, very long. It's very, very involved. Can we make that shorter and kind of easier? For the library preparation, that's kind of the next step. input DNA versus output data. So what we were testing before we kind of settled on the kit that we're using now, we tested out the PCR-based kit, but as I said, we found that it wasn't really amplifying very evenly. So we settled on native DNA kits, but that does mean you have to get more DNA to start with. And also with these library preps, some of them provide more data than others. With the ligation kit, you put in a microgram of DNA, but you get huge long reads because it's a different way that it actually sort of prepares the DNA. So again, this rapid kit is kind of a balance between it's relatively fast, as the name suggests, and it has a relatively good output, but it doesn't have the huge DNA input quantity that you need for some of the other kits. Then once you've done the library prep, you've got to base call it. This we were looking at is kind of accuracy versus speed. So this very much kind of revolves around computing power. There are 3 different versions of base calling that Guppy does. So Guppy is Nanopore's version of the base calling software. There are other versions as well, but it's got fast base calling, which is kind of the default setting, high accuracy base calling, and then super high accuracy base calling. And as the names suggest, one is faster, one is, you know, faster but less accurate, and the other one is much, much slower, but is much more accurate. When I say much, much slower, I tried to do this on my not particularly powerful computer, and it took literally a week to do the base calling. So fast generally seems to be good enough, but if you've got a GPU, it's probably much, you know, you could probably do high accuracy base calling in a relatively sensible time. Or if you have access to servers and you have programs, powerful programs on servers, then it might be worthwhile doing that. But again, if all you have is a relatively small computer, or just the Mark 1C, for instance, can you set that to high accuracy, or is it just too fast?
You can set it to high accuracy, yeah, but
it might take a while.
Just, yeah. If you're doing a run, it's just fast.
Yeah. Also, yeah. So I think there are options to change it to high and super high accuracy whilst you're doing the actual run, but that's going to take a huge amount of computing power and is going to then lessen your sequencing power. So unless you have a super computer, I wouldn't recommend doing them at the same time. What you might want to do is sequence and then individually base call it with high accuracy, because then your computer's not trying to juggle 2 things at once, and then it might give up and mind fries sometimes. Then we're looking at how we're actually analyzing the data. So what is it you actually want from the data you put in? And I think understanding your specific question that you're asking is really important, because it feeds into all of these different things. If, for instance, metagenomics, you're just looking, if you've got, say, a sputum sample and you want to know what might be causing somebody's illness, and you just want to know what species are in there, you could run that relatively quickly, because all you want to know, oh, that's an E. coli. You can run that in real time using Nanopore, have something called Epitome, which has a metagenomics pipeline. Within sort of 2 hours, you could easily identify, I've got E. coli, I've got Klebsiella, I've got, you know, whatever. But if you want to go down to sort of SNP levels and you're trying to identify 13 different SNPs, you know, or a difference of 13 SNPs, then you're going to have to get an awful lot more information, and the programs are different to kind of analyze that. So as part of this pipeline, we had a look at some of the different versions of the programs. We looked at Epitome, we looked at Microbe, if anybody's heard of that, and also TB Profiler, which are all kind of graphical user interface, so button cloud-based ones, as well as command line ones. And actually we found that generally TB Profiler was at the moment is the best one, because it's updated quite regularly. As Keith said, that's really important. Why do I think Nanopore is useful for sequencing TB? So I think Keith gave a fairly good overview of why Illumina is good for TB, but Nanopore has its advantages as well. As we were saying, it's got a really cheap in science terms, cheap initial setup. So for a MinION, $1,000. I mean, what's that, like 2 liters of some kind of reagent? So yeah, $1,000 is really cheap, although you do have to obviously consider computing costs and kit costs as well if you're carrying on. But obviously in terms of laboratory footprint and actual setup costs, compared to say a MySeq, which is maybe $50,000, it's much cheaper to sort of start. It has a smaller laboratory footprint, so a MySeq is kind of like that big, and MinION is like that big. So it doesn't take up much space in your lab, or you can, use it to do mobile laboratory stuff. You can go out to rural areas or wherever, the forest, wherever you want to go and do it. It's much better for sequencing on the go. I think this is where Nanopore really comes into its own, though, is the fact that these long reads can span what are traditionally very hard to read sequences. So as we've been saying, TB has a genome that is very GC rich, which can cause its own problems in terms of downstream applications. And it also has lots of these really big repeat regions. And if you have lots of really short ones, as Keith said, it might be quite difficult to find out where they fit in terms of the jigsaw of the genome. But if you have long reads, they might span all of that because, so as a general rule with this kit, your average read length tends to be anywhere between about 10 and 15 kb long, because when you add the transposase, which adds the barcodes, it actually sort of breaks it up. So this is why it suggests you have to have really high DNA in the first place. But on one or two of my runs, admittedly not TB, but on other things, I've got like 400 kb. Like one of my reads was 400 kb that went through. So long reads, I mean, that's like almost genome, isn't it? So that's that's a ridiculously long read. And obviously, if you had what is essentially an entire genome, you don't need to then worry about, assembling it. It's very obvious where it goes within the reference genome. So as Keith said, assembly is much, much easier with Nanopore. They both have their advantages, as he said, though.
That's like 10 full with genome. So it's
like
30 kb, yeah.
So as I say, it very much depends. I find with TB, it tends to break it down to be a bit shorter, and your average, so what's called your N50, is somewhere maybe between 6 and 12, maybe. With the new kit, it's better. But with, again, I feel like I'm picking on E. coli being the good boy. With E. coli, it tends to be more like, and with this new kit, when I did it, my average, my N50 was 16 kb, which is huge. So yeah, and the different kits will give you different size lengths. With the ligation kit, because it doesn't break it in half to add your barcodes on, what it does is it ligates the stuff on. It leaves whatever you put in, that is how long your strand will be. So you can get some phenomenally long strands if you use the ligation kit. But again, you have to have a huge amount of DNA to put in the first place, because you lose quite a lot in the process. Nanopore has relatively straightforward library preparation. We're going to go through it tomorrow. If you are sort of good at this, if you've done this a few times, this new kit has a bead washing step, so it does slow you down a little bit. But if you were doing, say, I don't know, 12 barcodes, you could probably have it all done within about an hour and a half, maybe, if you were like on it. We're going to be training, so obviously I'm not expecting everybody to be done in an hour and a half, but yeah, if you know what you're doing and you're just there and you're going through it, then I would say, yeah, with this new version of the kit, even with the bead wash, it's probably going to be about maybe 2 hours for a big set of samples. So it's quick and it's relatively straightforward. So this amount, actually, I'm going to scrap that. I think we can get it lower than that, because with this new kit, you can put far more on. And if we can get this new extraction technique to work that Nanopore have given us to trial out, I think we can get it down to around the same cost as we can with the gram negatives. So somewhere between maybe $50 to $60, depending on how you're extracting and how many you're putting on. So we'll start with $160 per sample, but I think we can get it better than that. I think we can improve on that. Problems. Nanopore sequencing, especially if you read the documentation and literature from sort of when it first started, generally does still have a lower accuracy than Illumina. So we need to make sure that we validate what we're seeing is what we're getting. And this is why that depth is really, really important. As I was saying, with Illumina, the sort of generally accepted minimum depth is 30. With Nanopore at the moment, it's 40. But there's new chemistry being released as we speak. The new kit, the new version of the kit that we're going to use is now out, but there's a really long lead time because it's literally just come out. And that's called Q20 plus. And that Q20, that's a quality score. So Nanopore has a threshold when it runs. You can set the threshold, but generally it sort of sets it at 8. So anything with a score of above 8 counts as a pass, and anything below that counts as fail. With Illumina, I think the percentage accuracy is generally sort of thought to be about 99%.
Now, yes.
With Nanopore, it obviously depends what you're doing. It depends on your base calling. It depends on the goods, you know, the quality of your input. It depends on all sorts of things. But I think at the moment with these versions of the kits, it's somewhere around 96, 97. It also depends on how good you, if it's your first time, it will probably be less. If if you're a machine and you've gone through it loads of times, then it's probably going to be better. So at the moment, Nanopore is generally thought to be lower, but you can find ways around it by essentially sequencing it more. And that should give you that confidence that what base you're seeing is actually what you're seeing. Nanopore kits generally need more input DNA, and this is mainly when I'm talking about the native DNA and the ligation kits. If you are doing a PCR-based Nanopore kit, then you don't need that much DNA. So if you're doing targeted, or you're doing, something that you only need a small section of, you can have essentially what you would need for PCR, basically. So I think it's anywhere 2 nanograms per microliter, so that's not a huge amount. But yes, with the native DNA, so you're not manipulating it in any way, you are literally just sequencing what DNA you've got. As we've been saying, somewhere between 50 and 400 for this kit we're using. And for the ligation kit, it's 1 microgram, so that's 1,000 nanograms, isn't it? So that's much, much higher. And then TB itself, as we've discussed, is quite difficult to work with. If any of you culture it, you'll know it's very, very slow growing and sometimes can be a bit demanding. We work with BCG a lot as well, and sometimes BCG just says, no, I don't want to grow. And so you left it for 2 weeks and it doesn't do anything. TB can sometimes be problematic in that way, but even if it's working, it's very, very slow growing. 2 weeks or so to get it growing from sort of maybe a sputum sample generally. As we discussed earlier, you tend to get a low yield from the extraction, but there are kind of ways around that. It has a high GC content that can interfere with how PCR and sequencing works, and it has many, many repeat regions. So you've chosen a really good organism to work on, is what I would say. We tested the different kits. So before we optimized our extraction method, we were generally getting somewhere between 5 and 100 nanograms per microliter. I'm going to say we erred on the side of 5 rather than 100, but that was too low for some of the native kits. So we started off by comparing the rapid PCR kit. So we were essentially just sticking all of our DNA in with a PCR with sort of the primers that come with the kit, and it amplifies sort of randomly. As we were saying, you can put in less than 2 nanograms per microliter, but it suggests somewhere around 2. But we found that amplification was not even a tool. So this is RPOB. So this is Artemis, the same program that Keith was showing you. You can see that our stacks are there, but you can also see that RPOB is not covered. So if your resistance marker, your SNP was on the other side where there is no coverage, you wouldn't, it just wouldn't see it. It's also not particularly good depth either, so we scrapped that. We then moved on to the old version, because this version wasn't out when we started this. The old version of the kit that we're using now, it required 400 nanograms of greater than 30 kb DNA. Ideally, normalizing it to femtomoles. So the reason we do that, if all of your DNA is all the same size, so it's all 60 kb or it's all 40 kb, that's fine. You can just do concentration. But if you have one that's 60 kb and everyone else is 30 kb, you've essentially got twice as much DNA in that sample. If they're all, if you've normalized them all to, as I say, 50 nanograms per microliter, but they're all 30 and that one's 60, that's twice as much information. And so you will find that that one will sequence better. So if you do have, and this is why, again, it's important to check the sizes. If you're getting differences in sizes, then you can normalize it to femtomoles instead of size and volume. So this kit gave us much better coverage, but it still needed improving. That's not the picture I wanted, but that's okay, because this picture, sorry, it's just changing these. That is the picture I wanted, actually. It's better than I thought. So this is the rapid barcoding kit. This is what you see. it's different as, as if you remember, I said with Keith's one, you see sort of lots of coins stacked on top of each other. It's the same with the PCR one because obviously it's shorter, but with long read sort of native technology, those reads are going to be really long. So they look a bit more chaotic. But as you can see, so some of them were quite good read depth, but others weren't so good. So then we moved on to this kit. So this is the one we're using now. It's essentially the same as that one, but it's got updated versions of all of the proteins and reagents. When we first tried it out, within 24 hours of sequencing for 6 isolates, so it wasn't very much actually. I think it would be better with more. So as I say, the more DNA you've got actually turns out better. We had more than 40 times depth, so that's the sort of minimum we would require for nanopore for all of the isolates. So that's just within 24 hours. So that's relatively quick. And after 48 hours, all of them were somewhere between 54 and 128 times depth. So that's much, much better than the original kit. And that is the picture I wanted to show you. You can see how much better the depth is and also the sort of coverage is compared to those two. So the coverage was much better on the rapid kit, but it doesn't compare to this one. This is much, much better. So this is the kit we're using at the moment. Very soon, hopefully we will try to test out the new chemistry version of this kit, but for now that's not really widely available, so we're going with this. But yeah, I'm very proud of that. I was really pleased when I saw that for the first time. But just as an example though, this is TB, this is Staphylococcus. So you can see how much more neat and actually I think the depth continues way above this picture. I just couldn't put it on. So it's just, we're working with an organism that's a bit tricky. This is the slide I wanted to show you. So this is an output from Minnow. So this is the software that we're going to be using. You can see, so if you're base calling at the same time, it will bring up this barcode chart. So you can see barcode 1, 2, 3, 4, 5, 6. Obviously, if you're not base calling, it won't be able to identify those. So you might have to do that afterwards. So how many have I got on there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I put 11 samples on there. Do you want to guess which one is TB? Yeah, that one? Nothing. I got nothing. And this is because, yeah, I know. Tell me about it. I could have cried. I did cry. All of these with the same amount of DNA, the exact same amount of DNA, because I normalized it to femtomoles. It's the exact same amount of DNA, but for some reason, TB's just like, no, I'm not gonna sequence. So this is why we're trying to optimize it to get it to not look like that. TB will sequence all right if it's with its own kind. If it's all TBs, then it sequences okay. But if you put it with something else, then they just outcompete it basically. So yeah, and as I say, we're hoping that this new version of the extraction protocol that we might get up and running, and also this new version of the kit, which seems to very much improve it, will hopefully mean we don't see that. We also tried ethanol precipitating after CTAB and bead washing as well, but it didn't seem to have that much of an effect. So we're hoping that this spin column based extraction technique might have a better effect. We've also found that, as I say, TB doesn't sequence as efficiently as other species. And we've been wondering whether, because it has this IgC content and the repeat regions, as I was saying, when the strand goes through, it leaves its other strand behind. I have a theory that that strand then, because of all these repeat regions, forms essentially big balls and clogs all of the pores. So we wanted to find out whether reducing the length of the DNA actually helped. To do that, we use something called a Cavaris tube, and it's a tube. Well, there's different versions of it, but the version we use is this tube, and you spin it at a certain speed and a certain amount of time to get specific kind of sizes. So most of our DNA was around 60 kb to start with. We sheared it down to 20 and 10. We then ran all of the samples for 2 hours on the sequencer, washed the flow cell and compared it to sort of our general 50, 60 original kb. The only caveat to this though is that Cavaris tubes are really expensive. They're about £25. So what's that, like $30 a tube? And actually, when you think that we've now reduced the cost of sequencing down considerably, that's like a quarter to half of the overall cost. So we did find that actually if you shear it down to 20, we got far more reads. But obviously you have to caveat that with the fact that actually the DNA is shorter and therefore you're getting less information out of each read. So the average length of the DNA after it had gone through sequencing was actually really, really short. Funnily enough, the largest that went in actually came out as the shortest from the sequencing. And then in terms of overall basis, which is essentially reads times length, again, 20 kb actually seems to work the best. But I think generally sequencing TB seems to be a trade-off between the DNA and, you know, the sequencing itself. So in the same way that CTAB is a trade-off between cracking open enough cells but keeping the DNA nice and neat. The same as can be said of sequencing. You have to kind of trade off between long reads and poor efficiency. But hopefully with this new extraction technique, that might sort of dampen that. So this is the final kind of extraction pipeline as we have it and as you are going to be learning it. Obviously, these things are, you know, increasing and improving all of the time. Nanopore and sequencing in general is just constantly evolving. But obviously we've got to have a cutoff so we can actually teach people. So our extraction, this was what we kind of wanted. And at the moment, our best bet is culturing and CTAB with the possibility of extra steps, but those include extra costs. With our library prep, we wanted some kind of, you know, we wanted to put as little DNA in as possible, but we also wanted to get the most out of it. And we found that this new version of the rapid barcoding kit seems to work best for that. In terms of base calling, this is very much a kind of what you have available situation, I think. Obviously, high accuracy or super high accuracy base calling is the best. But if your computer isn't powerful enough, then actually fast base calling with the most up-to-date version of Guppy, so the most accurate version of Guppy, generally tends to work, you know, pretty well. You might just have to maybe check your data when you get past that to make sure that it's definitely doing what you're sort of asking. But we generally found that when we analysed it down the line, that actually it was comparing quite well to the Illumina data that we had as a comparison. So yes, and then when it comes to the analysis, we found that TB Profiler compared to the other versions actually seems to be the best database and the most user-friendly database to upload it to. Or as I say, you can use the command line if you want. We're not going to go into that though, because that involves the Windows subsystem for Linux if you're on Windows, and then yeah, it's too much for one day. So TB Profiler on the cloud is probably the best way of doing it. So that's what we've got, and this is essentially what we are going through. So we've done the extraction. We've also explained about quality control. We're going to do library prep tomorrow, and then on Friday we're going to go through how to analyze that data. So it's very much a whistle-stop tour. Yeah, does anybody have any questions? Blown your minds with a lot of information today, I think. So we'll do some actual science this afternoon, I promise.
So you just concluded that you can only extract from culture.
So with this, so the sticking point for this is the fact that with these rapid kits, you need a certain amount of DNA. Yep. So 5 nanograms per microliter is the bare minimum amount of DNA that this kit will work successfully with. You might be able to do an extraction from a very high, you know, if somebody has really bad TB, you might find you can get 5 nanograms per microliter. But if your patient has low TB, or I suspect it's going to be quite difficult to get the amount of DNA that you would need to do it straight from sputum. We don't really do sputum extractions from sputum in our lab, particularly because we are a reference lab for clinical trials. So we tend to work mainly with culture anyway. So I don't know, how much would you say, do you extract from sputum? No. It's generally quite a faff, and you don't tend to get a lot of DNA out of it. Yeah. It also means, yeah, you're extracting specifically the thing you want, rather than, because if you extract straight from sputum, you're also going to be extracting anything else that's in there as well. So you might find that, so you can deplete the human DNA, which is probably 90% of your DNA that's going to be in that sample. Even if you then extract after that, you'll probably find that the, you know, just the internal flora of the lung is going to massively outweigh the TB. You've probably got like 1% TB or something, unless again it's a really heavy infection. It has been done and people are working on it for nanopore, but as a routine thing, I think it's still very much in its infancy. We can talk to Keith, but I think probably Illumina at the moment, because it requires so much less, because it's a, you know, an amplification based kind of platform, you'd probably find that might work better. But even then, I think with Illumina, it depends what kits you use, but I think ideally you want at least 1 nanogram per microliter. And that might be tricky if you're doing it straight from a sputum sample. I'm not saying it can't be done, and I know that people have done it, but in terms of an actual usable pipeline, I think in the moment it's not there yet, I would say. Probably in a few years' time, it might become the norm. So yeah, but yeah, at the moment you have to do culture first. But with this, you're culturing, you don't need to worry, like with DSTs, you would then have to culture with all of your drugs. You don't have to worry about that. You just do it straight from the original sort of culturing. We found that in terms of the amount of DNA you get out of the culture, we tested this as well. So we had samples that we'd grown up and we put them in midgets and on slopes, both LJs and 7H11s. And we found that if you put the same concentration in, the same amount into all 3 of them, if it's 2 or 3 weeks post flagging of midget, they all must grow roughly the same rate, because you get the same amount of DNA out. And actually, if you do it immediately after flagging, that's not enough. You don't tend to get very much DNA. The kind of optimum amount is sort of 2 weeks after flagging. So that's, well, it depends what you put in the first place, but that's what, 2 weeks, 3 weeks, 4 weeks after you've sort of started your sample, or, you know, subbed your sample. If you leave it for too much longer, you start to get to the stationary phase and things start to die again. And so then you start to get less DNA. So we definitely found that sort of the optimum, or the largest amount of DNA you can get out of TB tends to be about 2 weeks post flagging. So whatever the concentration is, they don't tell you, do they? Or do they? Is it something like 10 to the 6 CFUs, I think, CFUs? I think, so it's 2 weeks after that, basically, whatever that comes to. So you do actually need a fairly good amount, obviously. Again, if you don't put as much in, then you're not going to get as much out. Equally, though, if you put too much in, then you start to clog stuff up, and the amount of reagents isn't going to open the cells as well. So we've made this protocol that you guys have got because it works best on that amount of DNA. So hopefully you should get a fairly good amount. Again, even now in our clinical trials with our, you know, people who do this routinely on culture samples, we still get a very varied amount, and TB is just a very varied organism, I would say. So yeah, as I say, you picked a good one to work on, but it's going to give you some good answers to your questions, I think. So any more for anyone? Is lunch happening? Mm-hmm. Great. Let me stop this, please.